Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker 1969, wherein he suggested that the five kingdom classification Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification with kingdom Plantae, popularly known as plant kingdom. Okay, we must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell wall have been excluded from Plantae, though earlier classification placed them in the same kingdom. So, the cyanobacteria that are also referred to as a blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms and angiosperms under Plantae. Okay. So, let us look at classification within angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number, and shapes of leaves, etc. They were based mainly on vegetative characters or on the endoecium structures system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this natural classification system developed, which were based on natural affinities among the organism and consider not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. Bentham and Hooker. Okay. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. This becomes more important when there is no supporting fossils evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses the chemical constituent of the plant to resolve confusion and are also used by taxonomists these days. First one, algae. Okay, algae are chlorophyll bearing simple thyroid autotrophic and largely aquatic, both freshwater and marine organism. They occur in variety of other habitat, moist stone, soils and wood. Some of them also occur in association with fungi, lichen and animals, example on sloth bear. Form and the size of the algae is highly variable, ranging from colonial forms like volvox and filamentous forms like Eulothrix, Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation, each fragment develops into a thallus. Asexual reproduction is by the production of different type of spores, the most common being the zoospore. They are flagellated, motile and on the germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size, as in Eulothrix, or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in a species of Eudorina, is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as oogamous. Example: volvox and fucus. Okay, so here is the some figures of green algae, brown algae, red algae, volvox, Eleutheryx, laminaria, fucus, porphyra, polysyphonia, okay, dictata, as mentioned in the NCERT. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on the earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producer of energy rich compounds which form the basis of the food cycle of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloid water bodies holding water holding substances. Example: algine, brown algae, and carrageen, red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice cream and jellies. Chlorella, a unicellular alga rich in protein, is used as food supplement even by the space traveler. The algae are divided into three main classes: chlorophyceae, phaeophyceae, and as well rhodophyceae. Chlorophyceae. The members of chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage body called paranoids located in the chloroplast. Paranoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospore produced in zoosporangia. The sexual reproduction shows considerable variation in the type of and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eleutheryx, Spirogyra, and Cara. Now, Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitat. They show great variation in size and form. They range from simple branched filamentous form ectocarpus to profusely branched forms as represented by kelps, which may reach a height of 100 meter. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment, fucoxanthin, present in them. Food is stored as complex carbohydrate which may be in the form of laminarian or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by gelatinous coating of algin. The protoplast contain, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by holdfast and has a stalk, the stipe and leaf-like photosynthetic region, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellated zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common form are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. And now Rhodophyceae. The members of the Rhodophyceae are commonly called red algae because of the predominance of red pigment or phycoerythrin in their body. Majority of the red algae are marine with a greater concentration found in the warmer areas. They occur in both lighted region close to surface of water and also at great depth in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Gracilaria, and Zeridium. Bryophytes. Bryophytes includes the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. So these are the figures which is bryophytes. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sexual organisms in bryophytes are multicellular and the male sex organ is called antheridium. They produce biflagellated antherozoid. The female sex organ is called archegonium, which is flask shaped and produce a single egg. The antherozoid are released into water where they come in contact with the archegonium. The antherozoid fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not a free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division, meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are little economic importance, but some mosses provide food for herbaceous animals, birds, mammals, and other animals. A species of Sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses along with lichens are the first organism to colonize rock and hence are of great ecological importance. They decompose rocks making the substrate suitable for the growth of higher plant. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grows usually in moist, shady habitat and such banks of the stream, marshy ground, damp soil, bark of the tree, and deep in the wood. The plant body of a liverwort is thalloid. Example Merkensia. The thallus is dorsiventral and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverwort takes place by fragmentation of thalli or by formation of specialized structures called gemma, single gemma, or you can say gemma. Okay, gemma are green multicellular asexual bud which develop in small receptacles called gemma cups located on the thalli. The gemma become detached from the parent body to and germinate to form new individual. During sexual reproduction, male and female sex organs are produced either on the same or the different thalli. The sporophyte is differentiated into a food seta capsule. After meiosis, spores are produced within the capsule. These spores germinate to form living organism, free living gametophytes. Moses. The predominant stage of the life cycle of a mouse is gametophyte, which consists of two stages. The first stage is protonema stage, which develops directly from a spore and it is creeping, green branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They are attached. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoid. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a food seta capsule. The sporophyte in mosses are more elaborate than that in liverworts. The capsule contains spores. Spores are formed after the meiosis. The meiosis have an elaborate mechanism of spore dispersal. Common example mosses are Funaria, Polytrichum, and Sphagnum. So now pteridophyte. The pteridophyte include horsetail and ferns. Pteridophytes are used for medicinal purpose and soil binders. They are also frequently used on ornamentals. Evolutionarily, they are the first terrestrial plant to possess vascular tissue, xylem and phloem. You shall study more about this tissue in chapter 6. The pteridophytes are found in cool, damp, shady places. Some may flourish well in sandy soil condition. You may recall that in bryophytes the dominant phase in the life cycle is the gametophyte plant body. However, in pteridophyte the main plant body is a sporophyte, which is differentiated into true root, stem and leaves. This organism possesses well differentiated vascular tissue. The leaves in the pteridophyte are small microphyls, and in Selaginella or a large macrophyls as in ferns. The sporophyte bears sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones. Selaginella equisetum, the sporangia produce spores by meiosis in spore mother cells. The spore germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes required cool, damp, shady place grow because of the specific restricted environment and the need for the water for fertilization. And the spirit of living organism living spread of the living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids. The male gametes released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonia result in the formation of zygote. Zygote thereafter produce a multicellular well-differentiated sporophyte, which is dominant phase of the pteridophyte. In majority of the pteridophyte, all the spores of the similar kinds, such as such plants, are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro-large, micro-small spores known as heterosporous. The megaspores and the microspores germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophyte, takes place within the for variable period. The development of the zygote into young embryos takes place within the female gametophyte. This event is a precursor to seed habit, considered an important step in evolution. The pteridophyte are further classified into 4 classes: Silopsida, Silotum, Lycopsida, Selaginella, Lycopodium, Sphenopsida, Equisetum, and Pteropsida, Diopteris, Pteris, Adiantum. Okay, So now let's read about gymnosperm. The gymnosperm, gymno means necked, sperma means seed, are plant in which the ovules are not enclosed by any ovary wall and remain exposed both before and after the fertilization. The seed that develops post fertilization are not covered, that is, are naked. Gymnosperm include medium sized tree or tall trees and shrubs. One of the gymnosperm, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally taproot. Root in the some genera have fungal association in the form of mycorrhiza pinus, while in some other cycas, small specialized root called coralloid root are associated with antofixing cyanobacteria. The stems are unbranched cycas and branched pinus cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extreme of the temperature, humidity, and wind. In conifers, the needle-like leaves and reduce the surface area. Their thick their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm and the gymnosperm are heterosporous. They produce haploid microspores and megaspores. The two kind of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lacs or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspore develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cone or strobili may be born on the same tree, Pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated to form one of the cell of the nucleus. The nucleus is protected by envelope and composite structure is called an ovule. The ovule are born on different trees, okay, born on megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form four megaspores. One of the megaspores enclosed within the megasporangium develop into multicellular female gametophyte that bears two or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm the male and the female gametophytes do not have an independent free living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules born on the megasporophylls. The pollen tube carry the male gametes, grows towards the archegonia in the ovules and discharge their content near the mouth of the archegonia. Following fertilization, zygote develops into embryo and ovules into seeds. These seeds are not covered. And our angiosperm. Unlike the gymnosperm where the ovules are naked, in the angiosperms or the flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperm, the seeds are enclosed in fruits, and the angiosperms are an exceptionally large group of plants occurring in wide range of habitat. They range in size from the smallest wolfia to tall trees of eucalyptus over 100 meter. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes: the cotyledons and monocotyledons. Angiosperms, dicotyledons and monocotyledons. That's it. Thank you.